Hi, so my name is Fletcher. I'm from Bangor University. I'm a PhD student and a TA, but unlike my daytime TV counterpart, I don't solve murders in my spare time. Anybody who laughed has seen much TV. So I'm going to be talking about Cds1 today. So Cds1 stands for checking DNA synthesis 1, and acts at the S phase checkpoint. So during S phase, the DNA is being replicated. So the replication fork is travelling along, and then if it encounters a problem, it stop, and then we get recruitment of these replication checkpoint proteins. So we have Rad17, which is thought to facilitate loading of the 911 heterotrimer, and we have Rad3 in the Rad26 complex. So Rad26 is a regulatory subunit. So Rad3 phosphorylates a number of residues in this MRC1 protein which travels with the reprosome. But there's only two phosphorylated residues which are required for interaction with Cds1. And Cds1 can interact with either one of these. Cds1 interacts with the MRC1 at these phosphothreonine docking sites. And this orientates it in such a position that Rad3 can then phosphorylate Cds1. And this phosphorylation primes the molecule. And then two CDS1 molecules, two primed molecules, can then come together in a homodimer and trans-autophosphorylate each other. How they do this, I'm not quite sure, because if they're not active yet, then how do they phosphorylate the other one? So they just suggest that there's some magical transient activation of one of the molecules that can then sort of cause a cascade of activation. However, once they are activated, they can then go on to have their effector outputs. So these include things like maintaining CDC2 phosphorylation to prevent mitotic progression. It can downregulate HSK1 to prevent late origin firing, so you're not getting any more DNA being replicated in an unfavorable environment. And it can cause delocalization of certain proteins such as MUS81 and RAD60 away from this stalled fork to protect it and protect the genomic integrity. So if you look at Cds1, it's got three domains of interest. So N-terminally, it's got the SQTQ domain, and it's in this region that threonine 11 is phosphorylated by the Rad3. We then have this, it's a highly conserved domain, known as the FHA domain, or fork-head-associated domain. And it's this domain that interacts with that phospho-MRC1 at that stalled complex. And then we have the active domain of Cds1, so it's a kinase domain, and it's a phosphorylation of threonine 328 that activates the molecule. And you'll notice I've labelled all the methionines, so these are the sort of start codons that you can get the protein being produced from. So methionine or start codon 1 will produce the full-length protein. And what we found is alternative translation from internal start codon sites can produce smaller variants of this protein. So you can see here, this is just a Western blot. So this main band at the top, that's our full-length Cds1, so it expresses all the domains. What we've seen is that under certain stress conditions, so raised temperature, MMS, hydrogen peroxide, potassium chloride, you can see smaller forms of Cds1 being produced. And we know that these aren't just degradation products, because when we add cyclohexamide to the mix, which blocks alternative translation, you can see that these bands disappear. So we know that they are alternative translation forms. And through mutational analysis of these internal start codons, we figured out that this band here that I've labelled Cds1b actually results in just the kinase region of Cds1 being produced. So it's missing those two front domains, which would suggest that this shorter protein can act outside of that chromatin environment because it doesn't have sort of the FHA domain to interact there. But it also raises questions about could this kinase be active? There are certain conditions in which we don't see any inductions of variants, such as hydroxyurea and other drugs such as foreign-cure and camptothecin. Induction of the variants, though, is very rapid. So at 30 degrees C, where we're not seeing any induction, and then as soon as we switch the temperature up to 40 degrees C, you can see the variants being induced within inside 30 minutes, and they persist throughout the course of the experiment. So it's a very rapid induction. It's possible that this Cds1b, that shorter variant, is required for survival under certain conditions, although this isn't quite certain just yet. So in both acute and chronic studies, you can see that when we take away the Cds1, sorry? When we take away this Cds1b variant, so when we mutated that start codon, there's some susceptibility, and when we can only express that Cds1b variant, there's also susceptibility on exposure to hydroxyurea. But conversely, we don't see any susceptibility in OMS, and that kind of is confusing because we see induction with OMS, but we don't see induction in hydroxyurea, so it seems a little bit backwards. And Thomas sort of briefly introduced this before. What we've seen is an interaction with Chk1. So when we do size exclusion chromatography, they both come out in sort of the same fractions, indicating the part of the same complex. And you can also see that Chk1 actually gets pulled down with Cds1. So about 50% of it seems to be somehow attached to Cds1. And what's interesting, this is a summary slide now, so this is in the 30 degrees C. You see Chk1 is pulled down with Cds1. And this is the same with the hydroxyurea, so we don't see that association lost. However, then when we then turn the temperature up or expose the cells to MMS, what we've seen is dissociation of this complex. So we think that somehow the Cds1b is causing this dissociation. But just like nipple tassels, I know these variants exist, but I haven't found a use for them yet myself. So I just want to say thank you to Thomas and Christian for some of the data, to the rest of the group, and also I should say thank you to my sister for designing my Science Kicks Butt t-shirt.